So our company does primarily mRNA-based technologies in relationship to both IPSC generation and genome editing and cell-based differentiation, and that part is kind of new, using messenger RNA to direct stem cell differentiation. So just a little bit about our company. Our company is a 16-year company in San Diego that I founded in '99. We've always focused on technology development, and over the years we work on RNA action gene expression control. We also developed new fluorescent proteins and super resolution imaging, put them into various types of sensors. Those can be integrated into our stem cell derived cell models using CRISPR and other technologies. We also work on nanoantibodies isolated from llamas, and also, of course, what we're going to talk about, the CRISPR using ORNA and also IPSC generated with CGMP protocols. We own this current building now. We're expanding into a new building with CGMP capability of producing IPSCs, because our IPSCs has been licensed by ACT, now called Okada, using stem cell derived cells, RTEs, and photoreceptors to cure eye diseases. So first of all, this is how we use messenger RNA to create iPSCs. And this is showing, this short video shows how we started from fibroblasts, supplying the fibroblasts with a medium supplement composed of messenger RNA cocktail and transfection reagent. And by day 8, you already see most of the cells becoming stem cells. By day 9, most of them are indeed stem cells. And these cells have been, of course, checked by all sorts of means and also interact into various cells. Some of them are geared towards clinical trials, like I mentioned earlier. And the iPSC part has been published, put into current protocols, so you can look up online if you want to see the details of how we do this. Now, I don't need to go into details of any of these. The only thing I want to say is we focus on the messenger RNA platform delivering Cas9 cases and the BET Cas as well. So we actually started this whole Cas9 protocol in 2012 before the flurry of publications. And I remember after seeing the first science paper from the original group, put in SPR grant, and the reviewer was saying this won't work in the mammalian cells because of chromatin structure. That was very interesting back then. But later we got a grant and we started working on this. So here it shows just one example. I'm not going to a whole lot of details about how we do CRISPR now, but so far our platform has been recognizing 2 nuclease sites before and after the intended switch, and then supplied with a donor fragment, as you have heard a lot, so I will not talk a whole lot of details about this. Now, the next focus of our messenger RNA-focused technology is to use combination of messenger RNA and or novel differentiation protocols to derive cells, and this video will show you an example of how, in principle, we do that. So at the beginning, the cells were seeded as iPSCs, actually. They started from iPSCs. They were introduced with one messenger RNA. You can see the cells start to change morphology and attach. And there's actually neuron-looking things sticking out and form a complex. Later on, when we stain them, this is a 3-day, you can see network formation between neurons. And by day 6, you can stain positive for synapses. That is how we can derive neurons in about 6 days. And with a similar technology, similar methods, we were able to generate more specific neurons, such as the GABAergic neurons. This is a staining of tubulin and staining of the GABA transporter 1 protein. And similarly, this is a dopaminergic. And you have the TH staining in spring. By using a similar technology, we're able to derive neurons towards more specific types. Okay, so another example of using our iPSCs to make drug screening relevant cell types. And this is a typically known protocols in the field. To make exercise has been relatively difficult. One protocol is half a year, the other would be a couple months, and we can do this in a week. And that is on day 3, start to see hepatocyte-like cells. On day 5, they grow quite, exercise between cells. And day 5, they have this C-star shape and have some flu-like looking things sticking out. Of course, the most common uses marker for exercise is GFAP, and they're positive, they remain positive for quite a few passages. And similarly, this is hepatocytes. This is a public protocol back to Hamilton. There is a combination of various growth factors, switching medium, quite a few, later on, different growth factors than early on switch. So when we do it with one chemical to initiate differentiation, one transfection of one messenger RNA, and use another chemical on between day 5 and day 6. So on day 5, you start to see these hepatocyte-looking cells, and they still continue to grow, and showing phenotype that looks like hepatocyte progenitor cells. And when we stain with AFP, these are positive for many passages. Okay, so adipocytes is relatively quicker compared to the other ones by the published protocols, and sometimes involves sorting, sometimes involve other type of selection. But when we do it, we do even faster than most of the known technologies. So this is a 6-day protocol going from stem cells from IPSC to white adipocytes, and this is oil red staining. You can see it's really 100% cells are heavily, filled with oil droplets. And this is a different protocol. It's not just 2 days ahead of that. This is a different protocol. You can see the cells have different morphology, different reflection of light, and their oil droplets show the indication of their ground cell type of ground fat type of adipocytes, and also they have these dark droplets really are indeed oil droplets. And we just got this data last night actually. There is a positive staining on the uncoupled protein 1. This is kind of dark, but there is positive staining of those cells. Of course, those cells are still very immature, and they need to be further matured if we're going to see better staining along this pathway. So just to summarize, these are the things that we do in terms of cell differentiation for various drug screening or toxicity test purposes. So there's the metabolism group, the muscle cells, adipocytes, and there's the toxicity, hepatocytes, cardiomyocytes, neurons, and things like that. And after we have the cells, what do we do with the CRISPR technology and the cells combined together? One thing we're going to do is we will integrate genetically encoded sensors based on our own fluorescent proteins. Our company owns the brightest fluorescent protein in the blue, green, photoconvertible, and tetrameric red range. So we were able to create a lot of sensors, and this is one example in the background of such a sensor. It's called FPX technology, where you have one color before a calcium signal, you will have the second, so there's a switch, almost like FRET, but without the complication of the multiple wavelengths. In the FRET situation, you'll have a different color with internal control, dose ratio, and to reflect. And this is all the current genetic sensors are faster than before. So because we can integrate this through CRISPR into our IPSCs or IPSC, upstream of IPSC such as fibroblasts, or at any stage, we could create, ready-to-use drug screening or toxicity test cells that already have internal sensors and signals, and those are typically more consistent, and you can get the cells for the same assay basically based on the unlimited amount, because if it's started at IPSC stage, you can expand that forever. So in terms of format of assay, this is what we like to do, and this is called high content screening in collaboration with our partner Biosciences. And this is really a cell by cell, 100 frames per second type of screening, and each cell is analyzed and represented here. And we actually took on the job to do a blind test of longevity toxicity, comparing to in vitro, which is 65% match of the known toxic effects. 77% animal ex vivo, 95% animal in vivo, and with our platform, we got nearly the same as the actually in animal tests of this toxicity. So our cells and our sensors are basically geared towards this type of screening. Of course, it can be used in other platforms as well. This type of screening not only can give you really fast, like I said, each cell is measured. Actually measures the movement of subcellular targets such as a IRS1 fused protein, when it sees the insulin signal, it will migrate to the surface of the cell membrane. Towards the cell membrane, and that can be analyzed. The movement can be analyzed through the software. The speed is we can do a 3D4 plate in 8 minutes, analyzing each cell. And even with that, we can still have the high resolution. This is also very new data, showing staining of our cardiomyocytes generated through the method that I just mentioned. I didn't quite mention how we do the cardiomyocytes, which is also using this really fast protocol. And this is another commonly used marker for cardiomyocytes. And you can see the detailed structure. I think it's not really focused, but you can see like here, the whole cells actually throughout the cells. You can see this fibril structure and typical for the cardiomyocytes. It's another high resolution picture. And this is how we see the future of drug screening by having more information in really, really fast fashion. So the way we produce our assays, and we do, as our business model, do this kind of experiments, is we like to also sequence our cells, especially before we bank IPSC at all the bankable cell stage. We like to bank IPSCs, and also we like to always find a stage where this is a progenitor type of a property of any lineage, where we can expand to a really large amount and easily bank them. At that stage, we also like to see, we have been sequencing both their genome sequences and also transcriptome analysis, and we match that with the cells as a package for our pharmaceutical customers. So this is the summarization I have. We can get cells from different sources, patient-specific, disease-specific, as you have heard. We can do CRISPR at this stage, and then we can integrate the sensor that I mentioned, either to safe harbor or behind a natural promoter through a 2A peptide, basically all reflecting the real expression level of the target. We can create mutations, of course, and make them into iPSCs, as we can do this really efficiently. Doing a few hundred, few thousand lines is really not a big deal. You can see how fast we can do that. Also in multi-throughput way. We can differentiate them into various types of cells, as I've just shown, and that is all geared towards drug screening. Thank you.